Welcome back to the Plantopia podcast, the plant health podcast produced by the American Phytopathological Society. I'm your host, Jim Bradeen, a professor of plant pathology and associate vice president for strategy at Colorado State University. And today I'm delighted to welcome Juliana Gonzalez-Tobon. Juliana is a PhD plant pathology student at Cornell University, where she studies the role of small non-coding RNAs in regulating interactions between plant pathogens and their hosts. Juliana first came to love plant pathology as an undergraduate student at Universidad de los Andes in Bogota, Colombia. And Juliana has a very impressive publication and presentation record for an early career scientist. Today, we're going to talk a little bit about her current research, and we're also going to explore her motivation behind efforts to use social media to fight COVID misinformation, an effort that combines her unique research history and interests. Juliana, thank you so much for joining us on Plantopia.
Jim, thank you so much for that introduction. Super excited to be here. I've listened to all the episodes that have been out, so I'm just so happy and honored to be here.
That's fantastic. Thank you so much again. I really want to know a little bit about you as a person, how you got into plant pathology. I understand as an undergrad student, you started your studies thinking you wanted to work in the area of human genetics.
Yeah.
How did you end up in plant path?
Well, when I was, deciding what to do as a career and just picking an undergrad, program, I was very set on wanting to do something that will lead me to work in human genetics and human cancer research mostly. I had like a very strong personal motivation to do that. And in Universidad de los Andes, which is my alma mater back in Colombia, they have two programs. One is biology and the other is microbiology. And honestly, when I was picking, what to do, I had no idea what microbiology was. So I was like, well, let's go with biology, which is what I understand and what I know could lead me to what I want to do. During, the process, it was a 4-year program. And after 2 years, I realized that, yeah, I liked it, but I was much more of like a lab person than a, outgoing and 100% field person. And that's a little bit where the biology career was focused there. So I decided to do both. They gave you the option of mix and match programs. So I did both of them. And that's the point where I started to try, join labs to gain experience in what I wanted to do, right? In molecular biology, in research. And I joined a human genetics lab. And I was very close, honestly, to just do my dissertation there and just continue there. But situations in that lab led to me being in a moment where there was no really like an advisor that was available to guide me in what I specifically wanted back then. I didn't feel very supported there. And I did remember that I had just taken like a class that's called a sort of fungal biology or just fungi in general with Silvia Restrepo, who you might know for MPS. And I went to talk to her and I said, I'm not sure this is the lab for me. I'm not sure I'm going to have like the chances of research, but I do know I like pathogens. I do know I like microbiology and molecular biology. And I had just seen also a class called epigenetics. And I was like, this is amazing. I want to do everything in this in my life. And she was like, well, I love everything that you're saying and you're super welcome in my lab, but I don't work with humans. I work with plants. And I was like, what? And that was the moment I started to understand that, we have human diseases, we have human research, but then we have plant diseases and plant science research and the importance of that. And it all sort of matched, those moments in science where everything sort of comes into place in your favor. I was also starting to take a class basically called plant pathology, and just everything came together in my mind. And I was like, this is it. This is what I want to do. She opened the doors to her lab to me, and I did two undergrad theses actually right there, because I want to do one for the biology program and one for the microbiology program. And then I just ended up with so many questions that I stayed and I did my master's there, all in plant pathology. So yeah, that was the beginning of it.
That's amazing. The importance of good mentors and of those firsthand experiences really, really critical for so many of us in this field. You mentioned your master's, and I recently saw a preprint on the bioRxiv server that I think came from your master's research. The title of that is Evaluation of Small Non-Coding RNAs as a Possible Epigenetic Mechanism Mediating the Transformation from Biotrophy to Necrotrophy in the Life Cycle of Phytophthora infestans. So there's a lot of great information just in the title. And
yeah.
I think in plant pathology, many of us probably know what Phytophthora infestans is. But for our broader listenership, tell us a little bit about Phytophthora infestans and why we care about this thing.
Yeah, and that's perfect to actually continue with this story, because when I ended up in Sylvia's lab, she worked with different things, but her main focus at that moment was Phytophthora infestans, which, the common name would be late blight. And it's a very important disease, both in Colombia, where I'm from, and in the world, basically. I don't know if, a more broader listener has heard about the big famine that happened in Ireland, like 1800s, that basically wiped off 30% of their population. It was also one of the main drivers for a lot of people there migrating to the United States and just looking for, starting life in a different place. And that was because this pathogen is so destructive, so extremely destructive, that it basically was wiping off their potato crops, which was one of their main, food resources that they had there, and also just economy resource, because they were also growing and selling that. And this, it's interesting to think that this pathogen has been around for so long, and it's still a very big problem, and it's still something that worries plant pathologists a lot. So when I heard about all this story in Phytophthora infestans, and I had the opportunity to work in this lab that was really trying to understand it better in Colombia, to use information from the, United States lineages that we have here, and then understand what we have down there in Colombia, I was like, this is very exciting. So what I did, both I can mix sort of undergrad and master's, because it ended up being, all together in almost 3 different articles. I can divide it in 2 parts. One is the one that you're mentioning, which is tried to really understand the molecular interactions that this pathogen has when it's infecting, or when it is, just thriving in the plant. And in that sense, that's still a work in progress, the one you read. That's why it's still a preprint, while the others are already published. This one, we wanted to see if small RNAs, which are something that has gained more attention in more recent years, but back then, this was like 2015, 2016, maybe, it was not really much studied in this pathogen yet. There were papers, of course, but nothing from Colombia, nothing, done there. And I wanted to understand how it could impact how this pathogen attacks the plant. And that's where the funny words you were reading come from, biotrophy and necrotrophy too. Go ahead.
Let me ask a quick question though.
Yeah.
What is a small non-coding RNA?
Great question. Well, I think most people are familiar now, especially after the pandemic, with just the word RNA, right? And how it's this molecule that's very important in every single living being. And we have different types of RNA. This one, the small non-coding one, are very small. And when I'm talking small, are like, 20 base pairs, especially in Phytophthora, they were really small. That are really interesting because their sequence, just normal RNA sequence, matches places in the genome. And the reason why they match different places in the genome is because they will guide a whole protein complex to whichever location they match to. And then that protein complex will help degrade, for example, an RNA transcript before it becomes protein or before it produces a protein. So these, we all know the normal, molecular biology dogma, where we go from DNA, our genes, which are basically our instructions, right, in our cells or the pathogen cells. Then we go to RNA, which is this transition from the gene to the protein. Sort of, I explain it in more broader terms as like an instruction, as if you were taking notes from a book before you actually go onto something with that. And then the end product or the result, which will be the proteins. So the small RNAs are, an additional thing that comes into play in all these normal process, where they can help us regulate which of these transcripts or normal RNAs, as we mostly know them from, whether they become or generate protein or not. And that is one of the reasons why this is part of what we know as epigenetics, which I mean, maybe we can get into detail later, but in general terms, we have genetics, right, where you have your genes and your genes say exactly what they are meant to say. But then you have epigenetics, which are these other processes that happen in the cells and decide or regulate whether those genes are becoming protein, when are they becoming protein, and in which parts, in our case our body, in the pathogen's case, well, it's different structures.
Great explanation. I appreciate that clarity. So how is Phytophthora infestans using these non-coding RNAs?
Well, that's what we are trying to figure out, but I can tell you two things. For this preprint that we're still working on, Phytophthora does something that some pathogens do, but not all, which is it starts living in the plant, let's say a potato plant, and it starts feeding on it. It eats the living tissue, the leaves, the tubers, even the stems in some cases. And then at some point during its life cycle, it just makes a switch and it starts being really aggressive, killing all of it. So not keeping it alive, just really making the tissue become necrotic and death. And that's what we call necrotrophy. So that's why this pathogen is an hemibiotropic pathogen, because it goes from biotrophy to necrotrophy. Other pathogens are always necrotic, like the moment they get there, they just kill everything. Others are always biotroph, so like they get there and they maintain the plant alive to feed from that. And what we think, and we're trying to show with this paper, that I hope will be finished soon, is that maybe these small non-coding RNAs are helping it turn on and off certain genes depending on the moment of the cycle where it needs to be. So yes,
so the
that's what we're
pathogen
trying.
really needs to regulate its own genes, right? So that it's
Exactly.
expressing certain proteins at certain times in that infection process.
Exactly. And that's what, usually happens with pathogens. But I felt when I was starting in plant pathology that I wanted to understand that better, because pathogens don't work the same at every single moment. They have to adjust and they have to adapt to the environment where they are at. The amount of food they find, the type of plant they are facing, the variety that they're facing. So there's so many different things that, yes, we have explanations for some of them that are very clear and like are very genetic based, but there were others that seemed to me, at least with Phytophthora, didn't have such a clear genetic explanation. They might have just a regulatory explanation behind them.
Yeah, it really strikes me. You mentioned that the Irish potato famine or the European potato famine of the 1840s. It really strikes me that we as a research community have done a lot of work on Phytophthora infestans and the potato late blight pathosystem. But we're 180 years almost out from the Irish potato famine, and there's still so much to learn about about the pathogen and the host and their interactions.
Yes.
Really exciting work.
Yeah, and that actually gives me a little bit of moment to tell you very quickly, and you can then decide if this makes sense, um, about another project we had during my master, which is actually already published. So this one you guys can read more easily. And it all stemmed from a mistake, if you want to call it like that, that happened here at Cornell in Dr. Bill Fry's lab years ago, like 7 years ago. And it was because, as we have been saying, since Phytophthora is so aggressive and so difficult to manage, one of the things that the community has done and the plant scientists have done during these years to control it is just use fungicides and just chemical compounds to control it. And that's what mostly people use in the field and growers use in their farms. So this lab, Dr. Bill's Fry lab, who has done amazing work in Phytophthora for so many years, they were testing different, fungicides, and specifically one that's called mefenoxam, very commonly used here in the US, and different concentrations of it to see, which ones of the lineages that they had in the lab, mostly from New York State, were susceptible to the fungicide or resistant to it, and in what concentrations. And imagine they were testing in a plate which zero fungicide, so the pathogen can live happily there, and then a meat concentration where it was enough to control its growth, but it was not enough to fully wipe it off, and then another one with a lot more fungicide to the level where it's just lethal to it. And they were just making routinary transfers from one to the other, and someone just messed it up in the sense that instead of making the transfer from the original one without fungicide, they made a transfer from the mid concentration, so the one that regulated it but not killed it. And then when they passed it to the same set of concentrations again, the pathogen was able to grow in all of them, even the lethal one, as if there was no fungicide there. And this is one of those things in science that sort of comes by serendipity, and you're like, oh, I thought this was a mistake, but maybe no, maybe it's something. Let's make an experiment, right? Let's plan an experiment to test that. And when they tested it, it turned out that it repeated, which is what we're looking for, right, as scientists, to see if things replicate and happen again. And it happened in different lineages. It happened in different times that they repeated it. That ended up generating a paper in, I think it's Phytopathology 2015. And in that paper, one of the authors is Juliana, who was in that moment a PhD student in Dr. Fry's lab. But then she went back to Colombia because she was originally from Colombia, and then ended up being my teacher of plant pathology. And when she told us about this, I was like, that is amazing. She also said, after they were calling it acquired resistance. If you put it back in media without the fungicide, it progressively loses that resistance. So it's not like the common fungicide resistance that we already know of, where you gain it and like that's it. The population is already resistant, nothing to do.
So that suggested it wasn't a genetic change.
Exactly.
But an
That suggests
epigenetic.
that it was a genetic change, so an epigenetic one. And that was one of the main drivers that I was like, I need to study this. And then the other paper I was mentioning, which was actually published last year in Plant Disease, we made all these set of molecular biology techniques, including small RNA testing, as you were mentioning earlier, to try and understand what are the things that are changing in the pathogen when it gets resistant and then when it goes back to sort of losing that. And honestly, that's one of the papers that I'm most proud of, just because it was so much work, so many years put in that. But it's a good way, and I think it's a very beautiful way to sort of show how pathogens are not static things, they change. And part of the challenge for us as scientists and plant pathologists is understand how that change goes and how we can play with that to our advantage.
I really love that. And it makes me think of the many times I've talked to different plant pathologists working in different pathosystems where, essentially they describe the plant pathogen growing in culture and it getting kind of lazy. It
Oh
becomes
yeah.
a less well adapted to the host. And I think what you're describing is a really good case of that, and really interesting that you've been able to pinpoint an epigenetic origin for this. I think it explains a lot in our science.
Yeah.
And I wanted to mention that on the plantopiapodcast.org, which is the official landing page for this, we'll include links to all of the papers that we've referenced today. So it sounds like you had a really incredibly productive and exciting master's program. And now you're at Cornell as a PhD student. I guess, first of all, how'd you get to Cornell? Why Cornell, and what are you doing today?
Yes. So, I've mentioned my two mentors from Universidad de los Andes, Silvia Restrepo and Joanna Danies. They were both 100% for Cornell. Joanna was, as I said, a PhD student here, and then Silvia did her postdoc here. So when I was finishing my master's, I sort of said, I want to stay in academia. I feel like I want to keep researching, but I'm a little bit lost as to what to do now. And they both say, well, just go and do a PhD. And I said, I have no money to do a PhD. And they said, usually nobody does. That's why we find for, funding. That's why we look for places that have programs for funding. And they said, Cornell is an amazing place. So in 2017, which was between the end of my undergrad and the beginning of my master, when we had already talked about, future in research, I had the opportunity to come to Cornell during the summer. Basically, it was from June to August, and just be an intern in one of the plant pathology labs. That summer, I was part of Dr. Keith Perry's lab. And it was a little bit different because they worked with viruses, but also small RNAs. In that case, they used small RNAs to understand which viruses they were finding in vines population here in the Finger Lakes, which is super important in this region for wine production. And so that was an amazing experience, not only in research, sense, and I learned a lot from them, but also just in going outside of my country, seeing how science was being done outside in the US in a gigantic institution as Cornell, and figuring if it felt like a place for me as like long term. And it did. I came back home feeling that I didn't want to do long term research here, and I guess I just tried to come back as much as I could. The following summer, which was when I was 100% into my master's, we realized that it was easier for me to come here and do some of my experiments in Dr. Fry's lab instead of like importing everything we needed and all the reagents and everything to Colombia, which is one of the challenges of doing research in smaller countries. So I did. I came here. I just confirmed that this was the place for me, and as soon as I came back, I applied to start my PhD. In the next fall. I was a little bit reckless in the sense that I only applied to Cornell. I then realized when I was invited as a recruiting student that all the other people that were here with me had applied to tens of universities, and they were like, which are your choices? And I was like, I only applied to Cornell. Like,
I like that confidence.
I don't know, I think it was a mixture of confidence, but also just like I didn't know. I was not aware that you had to have options. I was sort of like, if Cornell says no, then I'll just stay here for more, for longer and look for something else to do here. Um, but fortunately, they said yes, and I came and started my PhD in 2019, right before the pandemic hit.
That's great, and Cornell certainly is a great institution. What's it been like, though, as a graduate student in this pandemic period?
It has been challenging, I can say. I started in June, so that meant that I had the summer and the fall in sort of like a normal campus and a normal situation. I went to classes. I just normally what you do as a first year PhD student is like go to classes, start thinking about your research, do a small projects in the lab, and just get accustomed to it and to the people. But then as soon as the spring semester started, things started to look strange, and news kept coming of these virus that was around. Um, and then mid-March, there was just one day that Cornell sent an email and said, we're gonna bring the spring break closer, and we're gonna send the undergrads home. If you're a grad student, of course you can stay, but you cannot work in the lab until further notice. And that's it. And I guess, as probably everybody that is listening to this is, felt very scared. In my case, also, I think I have the additional challenge of being an international student. And I felt like I had just moved here. I didn't even enjoy a full year. And I was not sure if we were going to be able to stay or what was going to happen. And I say we because my husband moved here with me. Like I moved my whole life here. It's not that I just came myself. Um, but I think my advisor, which is Dr. Melanie Filatrud here at Cornell, she was super flexible. She was super understanding. And she said, well, we're just going to make the best of this situation. Um, we were not able to go back to the lab for about 2 or 3 months. So during that time, I did a lot of bioinformatics. I guess I benefited from the sense that I was just starting, and I was in a stage where I was planning most of what I was going to do. So, having time to sit down, look at the data, go into the genomes, download things, and start just thinking of what I wanted to do in the lab, but sort of doing like a pre-approach from my computer, that was very helpful. So when I came back into the lab during the summer, I felt I was more productive just because I had so much time to think that through. But after that, it has just been a challenge of like, moments when we were allowed to go in the lab. We had restrictions of how many people would be there and at what times. So you had to plan your experiments really well so that they fit in the time that you were able to go into the lab and not lose any time. And now that that's much more relaxed, I feel I have not stepped back from that. So when I go to the lab, I'm like super concentrated, and I do all these things. And then I realize, well, I can take it more slowly. I don't need to get out of here in like 2 hours. So yeah, but I think the biggest challenge, more like aside from the research, was just not having an in-person community around when you're such a young student. Also, my cohort was very small, so we didn't really have other people to rely on. I mean, you could talk to people on Zoom, but it's different. Um, and it's just the difference of then going back and seeing these younger cohorts being able to meet in person and like do take all their classes in person. I think it was difficult, but I don't know. I just think we made the best of it.
That story certainly rings true to me as well. You're describing that sort of the social aspect of science, and just despite how science is often portrayed in popular press or movies, there are really critical social communal aspects of science. Learning from each other, supporting each other, getting to know each other. That's really very critical, and it's certainly something that has been a challenge, particularly during the lockdown phase of this pandemic. I'm glad you've come through that period well, and I hope that for your sake and as well as all of ours, that the worst of that is behind us.
Yeah.
Could you tell us a little bit about what your PhD research is at this point?
Yeah. So, I came to Cornell with this experience in all my seats, because I have worked with Phytophthora so much. The scenario here at Cornell was a little bit changing in that moment. There was no lab that was working exactly what I've done before. And that was actually really great because I just wanted to use all that experience in molecular biology and just maybe apply it in something different. Um, I chose Dr. Melanie Filatrud's lab. She works with bacteria in general that affect plants. She had a very strong focus on Pseudomonas syringae, that, as you may know, is one of our model bacteria for plant disease, and that has been researched so much by so many people. But then more recently, she has included in her program another bacterium that's called Dickia dadanti. It was formerly known as Erwinia chrysanthemi, and I mentioned that because I know a lot of, seasoned plant pathologists are like, Dickia what? Like they recognize the old name, of course.
And this is a soft rot pathogen of
It
potato
is a soft
tubers?
rot pathogen. Yes, it is very closely related to Pectobacterium, which you might have also heard of. And yeah, its name was recently changed 2005. It became important, I guess, more important in the US and the Eastern plant pathology industry and research area, starting in 2018, 2019, because there was an outbreak in basically that affected basically all the states that produce potatoes. And people were like, what is this? Where is this pathogen coming from? Is it Pectobacterium? No, it's not. It's different. What is it? And a lot of, research institutions in the East, including Cornell, decided that they needed to, give a hand and start understanding this pathogen as well, which curiously has been more studied in Europe because it has been generating problems in Europe for a longer time. The species that we have here and there are a little bit different, but just the general genus has been widely recognized in Europe earlier than here. So my PI included Dickia in her work. That meant that she included potatoes in her work, and that was very attractive to me in the sense that I could both do, molecular biology and epigenetics, but also continue with the crop that I've been working with. And potatoes are not only one of the 5 major crops grown in the world and are exceedingly important in the US, but they are also very important in Colombia. So I felt that it was also a way to maintain things that are going to be relevant from my, for my home country. So so what I'm doing with Tikia, going back to that, is a very molecular based research as well. I am trying to understand how it senses its environment. So as I mentioned, pathogens have to understand what's going on and then respond to that. And one of the ways they do that, and mostly bacteria, is a process called chemotaxis. And it's just basically sensing chemical cues from the environment and responding to it. Those cues can be, plant compounds, defense compounds that the plants are generating when they're being attacked, and then the bacteria responds to that. So I'm understanding how that process works, but then I'm also understanding if there's again an epigenetic mechanism behind that will help them be quick and reversible. That's the 2 things that are most important in epigenetic mechanisms, that they are very quick to do, quicker than just, gaining a genetic mutation that gets fixed over time. And again, reversible. And we're looking specifically at a group of genes that are called methyl accepting chemoreceptors, but we call them MCPs for short, that are mainly required for that sensing process that I was saying. And curiously, Dickia dadanti, which is one of the species that we're working with, has so many of them compared to, closely related pathogens. And the question starts where, why does it have so many? Why does it need so many? And how is it regulating that many genes that could be doing the same thing? Or are they not? Maybe they're doing different things. So I still don't have answers, of course, because I have 2 more years to go here. But we do know that these genes have a very long upstream region, so right before the gene. It's a region that was considered to be empty, like no genes were there. But previous people from my lab realized that that region is actually being transcribed, and it's being expressed in some sense when the bacteria is infecting the plants. And that just made me think and made us think, well, there's something there that is not empty. What is it there? And up until now, we know that the structures that we're seeing there very much resemble small RNA structures. And we're trying to understand, which small RNAs are there. Are they actually regulating the genes that are right next to them, or are they doing something different? But we do know there's something there, and we're trying to get a hold of it. And if we do understand that, then potentially can use that to control how much these bacteria senses its environment. Of course, that's a super long-term goal, but everything in molecular biology starts like that. Like you have to look at the little details before you think of the bigger thing where you can apply that.
That is super, super exciting work. And I really look forward to seeing where this research goes. I think it has broad implications for our basic biological understanding of plant microbe interactions, but also disease control, as you sort of reference.
Yeah.
Very, very cool work. I want to take a moment, though, and pivot to a related topic. You're something of a rock star on Instagram. And in fact, you've got a really robust social media presence. You can follow Juliana on Instagram, on Twitter, on YouTube, and on TikTok, all under the handle EpiPlantPath. That's EpiPlantPath. I want to talk particularly about your Instagram account. You've got almost 10,000 followers. Your Instagram account is in Spanish.
It is.
And really is a professional scientific communication account. Tell us about that account, what you're putting out on Instagram and why you're doing it.
Okay. I'm going to ask you a question first. Do you have group chat with your family? Like with your extended family?
Not really, I don't.
Okay.
We text once in a while, but we don't routinely do that, no.
Uh-huh. Well, the reason why I'm asking that question is because to explain how this Instagram started, I need to make a point regarding how my roots and my Latin American culture are somehow different in some senses to the way maybe people here in the US relate to their families. And that's because for some reason we do have very big family group chats. That's a concept that has been hard to explain to my friends here in the US. They're like the same as you. They're like, what? I mean, I text my parents, maybe my grandparents, but every now and then I don't listen, hear from them every single day. And maybe people that are listening and are also from Latin American background or even a Hispanic background can relate in the sense that for some reason in our culture it's very common to have huge family group chats where things happen every single day and they share things every single day. And in my family group chat from Colombia, there's almost 50 people there, which is a lot. And they share everything they see, they share everything they consider relevant. And you might imagine that when the pandemic started, they just started sharing every single thing they saw in social media, everything they got in their, WhatsApp chats from friends. And I mean, that was fine. They were just trying to socialize what was happening, right? Relate to what was happening. But early 2021, when the vaccines started rolling out here in the US, or at least the first people being vaccinated started going to the news, I just started having to deal with messages every single day from my family that were 100% misinformation. There were things like, these vaccines are going to kill us, or what is it that they call RNA? What are they putting on that? What are they getting that into our bodies?
Am I correct that most of the folks on that chat, most of your extended family are not scientists?
Exactly. Yes, there are maybe two scientists, and the rest are just general, audience people. Like they do other things in their life, and they were very scared. And I think that's the main thing that made me feel uncomfortable with that. I was like, to me, seeing and reading about these RNA vaccines was like the realization of everything we've been talking about, doing very much molecular research and then making it be useful for the world and for humanity. And I was like so excited about them, about understanding how they work and why were they going to be rolled out in such a massive sense. But then I went to that chat and I saw how my aunts and uncles were extremely scared of just conceiving the idea of getting vaccinated with that. Um, so I told my husband one night, like, I'm gonna do something about it. I might just post something on my Facebook that I know they read and explain what is RNA very simply. Why is it being put in the vaccines? What is it doing in our body? And why is it not a reason to worry? Instead, it's a reason to be very happy about it. And my husband said, I think a video makes more sense. A video gets more people's attention. And I just grabbed my phone, like literally just grabbed it, sit down, talked um, by like 8 minutes in Spanish. And it was directly talking to my family. I sort of presented myself at the beginning, like introduced myself, just because I thought, oh, maybe they send it to their friends. So just so they know that I'm not like some crazy person trying to explain RNA. Um, and I finished that. I sent it to them in this group chat. And then I thought, well, it wouldn't hurt to just post that in my social media, which at that moment I had like 50 friends on Instagram, like just my friends. But I thought maybe they're having the same problem with their own families, and maybe it works for them to share this. I have to clarify that in that moment there was no information, like accessible about the vaccines in Spanish. There was already things in English, and usually the CDC makes a really good job putting out in their webpage explanations that are very simple. But in Spanish, we didn't have anything like that. And I just did that, left my cell phone aside, and continued my life. And the next day, it was crazy. It had exploded in Instagram. It was having so many views. I was getting a lot of people following me, and I was like not really understanding what was going on. But then by reading their messages, I realized they were all needing to hear that from somebody in Spanish, but they didn't have it. They have nobody telling them that. They were sending them to their families. And soon, what used to be a misinformation chain going between group chats in these population changed to be a chain of this video explaining vaccines very simply and why RNA was not going to kill us. So that was a moment very, it's a stressful, but it was very exciting to see that the way I had phrased things was resonating with people and was making them understand that at least just give them a second chance, just give a chance to understand the vaccines and not say no automatically. And just I feel it was just like a snowball that kept growing, and I kept realizing I could explain some other things, everything in the level of what people will normally just learn in high school, but maybe they don't remember what is DNA, what is RNA, what is a protein, what are our cells, how do they work. So just very simple things that if you don't work in science and maybe you don't have a passion for science, well, you just saw that in like fourth grade biology and then forgot it. But now it is important again because you're deciding whether to get vaccinated with something that will have a just make its work in a set of your cells. So yeah, I think it just kept growing. It kept having a really good response from people, and after that it just transformed into a more general science communication space in Spanish, just because there's not much available.
Oh, I really love that. Thank you for stepping into that space and using your scientific understanding, your biological background to help others. That's huge, and that's what Plant Path is all about too. And it's wonderful that you were in that place and really had the foresight to invest in this. I guess moving forward, how do you see social media fitting into your professional persona?
Yeah, that's a question I've asked myself for this whole year and a half. But I think I've come to some things that I value a lot from it. And one is that as scientists, we sometimes get really concentrated on only what we do, specifically for example, our pathosystem, our specific interests. And having a place or having a community that sort of moves me to read about other things, not only what I'm doing and what my really, close community is doing, but what is happening in, cancer, um, animal research, all these other things, and picking things that are important for people to understand, and that because they are impacting their own lives. Like in that moment it was vaccines, but it can also be gene editing, it can also be GMOs, climate change. Like there's so many big topics that have a science background behind it. So I feel like it's been one of the reasons that has brought me to a moment in my career where I feel very versatile, in that I love what I do, I love my research, and I see myself going forward in that direction. But I have like these um I don't know if it's like an alter ego, like another side of me that has a really nice community and can interact with it. And I think the other really valuable thing is that about 82% of the people that follow me on Instagram are women. And they are usually aged between 18 and like 38, 40. And from conversations with them, from reading their messages, I found out that most of them are just either already in science and they're doing their own PhDs, their own masters, their own undergrads, or just thinking of going into science. So it has also combined not only the idea of talking about science to a broad audience, but showing other women and other girls especially how it looks like to be a woman in science, and how it looks like to be day to day. Because sometimes you have like this really unclear idea of how it will look like when you go to do your PhD. But if you just open your Instagram, and among all the thousands people you follow, there's a few that show you like, oh hey, I'm doing a PCR today and this is what PCRs look like. um, so that has been rewarding, but at the same sense it has generated a bigger community for me that I do expect will help me in sort of my own academic path, to have these connections, having both my connections here in the US, the people, my close community, APS, that has been 100% supportive with me ever since I was an undergrad, to now, and then this other community that's all over the world, but it's just opening doors, elsewhere. And I feel like I'm opening one door here, which is this is what it looks like. If you feel like pursuing it, just this is an example. It's not going to be 100% like that, but just so that you have an image of how it is.
Fantastic. That really, it's exciting. There's so many scientists who are using social media these days, and I think you're doing it exceptionally well. Thank you so much for what you're doing in that space. And thank you so much for being our guest today on Plantopia. It's been a real pleasure to have this conversation with you.
Thank you, Jim, for having me. It was a pleasure to be here, and thank you all who are listening to us today.
And we just heard from Juliana Gonzalez-Tobon from Cornell University, talking about her research on small non-coding RNAs and the role they play in regulating plant pathogen interactions. We've also talked a bit about her social media presence and what she's doing to fight COVID misinformation and really raise the profile of science understanding and communication more broadly. I'm Jim Bradeen, the host of the Plantopia podcast. Plantopia is a production of the American Phytopathological Society. Thank you very much for joining today, and we will see you next time.